Hello, and a very warm welcome to everyone joining us for today's webinar titled Fast and Reliable Automation of the NEBNext Ultra 2 FS DNA Library Prep Kit for Illumina with Firefly. My name is Ellen Sims, and I'll be moderating today's session. I'm delighted to introduce today's webinar speakers. We're joined by Paul Lomax, the head of Genomics, and Maryia Karpiyevich, who's a senior applications development scientist at SPT Labtech. In this webinar, understand how to scale and automate library preparation workflows while maintaining quality. Learn more about Firefly, a new liquid handling platform that combines pipetting, dispensing, shaking, and incubation. And gain insights into how Firefly was used to generate libraries using the NEBNext Ultra 2 FS DNA Library Kit for Illumina. Following on from this presentation, we will have a question and answer session. So if you have any questions for our speakers, please feel free to submit these at any time during the webinar. There will also be some poll questions during this session, so do keep a lookout for those. So without further delay, I'd like to hand over to our speakers for today's presentation, and I'd like to thank them for presenting to us today. Please go ahead.
Hello, and thanks for joining the call. Today, myself and my colleague Maria are going to be talking to you about fast and reliable automation of the NEBNext Ultra 2 FS DNA Library Prep Kit for Illumina with the Firefly. To get started, I'm going to talk you through some basics of the products, just to give you an introduction to Firefly if you're not already familiar with it. And then Maria will actually be taking you through the details of the application. The Firefly was launched in February last year, so in 2022, and it's a very compact but comprehensive liquid handling system, which incorporates a number of different technologies in a very small footprint. You can see the dimensions in millimeters here, which actually just show how compact the unit is. It incorporates a dispensing system on the right-hand side of the instrument. You can see the 3 syringes, which are used for non-contact dispensing, which I'll go into in a little bit more detail shortly. And then on the left-hand side, you have a 384 tip pipetting head, which utilizes disposable tips and can also work with a range of plate formats from 96 to 384, and also has a gripper tool, which is able to manipulate plates on and off things like magnetic blocks and so on. Again, I'll go into more details on this shortly. It features 2 moving decks, each with 8 plate positions. So 8 positions for SBS format plates and racks, and also for disposable tips and so on. And then it also features working modules below the deck of the instrument in terms of incubators and shaking. So it really utilizes the space well by use of vertical space with 2 decks, one above the other, and then below deck positions as well, which is why we can fit so much capability into such a small footprint. It also uses a touchscreen controller to drive the instrument, and you can also author protocols and write new protocols offline on a PC and then send them directly to the machine, either via the cloud or a network and so on. Again, this helps keep the footprint small by not needing a PC at the side of the system. So just to look in a little bit more detail, and to start with the liquid handling capabilities of the pipetting head. The pipetting head is an air displacement system. It is capable of loading 384 tips or 96 tips, and also strips of 8 or 16, so that you can work one column at a time in either 96 or 384 well formats. So it's very versatile, and it can switch between all of those on the fly. So within a single protocol, you could use a combination of 96 well plates and 384 well plates, and also work across plates with strips, or just using partial plates if you got lower sample numbers. The tips are loaded from auto load cassettes, and the tips come preloaded in the formats of either 96 clusters or 384 clusters, or the individual columns of 8 or 16, and you can just slide those in for as many columns as you require in the protocol. So again, just kind of highlighting the versatility. The volume range is from 0.5 microliters to 125 microliters, which is reduced slightly with filter tips to 100 microliters. I'm just going to show you a few of the properties of the system, with firstly starting with the tip change. So here you see the it's actually unloading a set of tips that is just used. So it's actually unloading the cassette, and then it's moving to the back of the instrument to actually pick up a clean set of tips for the next step in the process. So it's very quick and easy to exchange tips on the fly within the protocol as you program it. So that's the first one. The second one is actually showing you now with a set of 96 tips loaded, actually doing a variable mix. So here we're mixing at a moderate speed, and we're aspirating low and dispensing high, which we find gives an effective way of mixing. And all of this is easily within control within the software, the speeds that you aspirate and dispense, the heights and so on. So you've got a lot of flexibility over your liquid handling steps. And then here we actually see it just using a single column of tips. So here it's using 96 tips to actually aspirate across a 96 well plate. So here it's actually being used to pool samples. So aspirating an aliquot from each column, and it will work its way all the way across that 96 well plate, and then ultimately dispense them into a single column of another 96 well plate, so that you've pooled 12 wells into one, and then a user can come along with a pipette and just put those down to a single tube or well. So it's a very versatile pipetting system, and as I mentioned already, it can switch between these formats on the fly. So now moving on to the dispense head capabilities. So on the right-hand side of the machine, there is a non-contact dispensing system that has the option of 3 or 6 heads. So when you buy the system, you can choose to have with 3 or with 6. Each independent syringe can operate from 200 nanoliters to 4 milliliters, and it works in a non-contact format. So the syringe flies above the plate and dispenses non-contact into each well as it goes by. For those of you familiar with our Dragonfly system, it uses the same technology and the same reagent reservoir options. And it uses a positive displacement syringe. So in other words, there's a plunger in contact with the liquid, so you have no air gaps. And what this means is we're able to dispense both very accurately, but also a wide range of liquid class types, from very volatile to very viscous, and even things like beads with particulates and so on. So it genuinely is liquid class agnostic and can do things that we often would struggle with the air displacement pipetter that I showed you just a moment before. It's also able to work across all common plate format types, and for these genomics applications, typically 96 or 384 well plates. We can either use each syringe individually for a different reagent type, or we can use multiple syringes for the same reagent for speed. So in the case of using, say, 3 channels for the same reagent, we can dispense 5 microliters across the 384 well plate in less than 1 minute. So it is very fast. Just show you a couple of videos. So the first one is just aspirating, in this case, in a single syringe. So the back left syringe has been filled with beads, and it's now just dispensing those beads across this 96 well plate, 1 well at a time. And even going 1 well at a time, you can see that this is quite a fast procedure. So you can get those beads dispensed very quickly across the plate. And these are typically quite difficult to pipette with standard systems. So it really is a good use of this technology. The second video, here we've actually already aspirated in 3 syringes, and now it's dispensing across a third of the plate with each syringe. So they're all working in tandem with each other, but you get obviously a much faster operation, 3 times faster than if you were using a single syringe. But it's entirely your choice within the programming how many syringes you dedicate to each liquid type. Just a couple of other things to give you a sense of other things that the system can do. So here, this one is just showing the grippers in action and moving a plate from one of the standard deck locations and then onto a magnetic block. So you can see the magnetic block just standing slightly tall there behind those two plates, and it's just moving the plate into position to pull beads down to the magnet. The second one actually just gives you a sense of how strong the grippers are, because now we're actually going to move that block. So within protocol, you might want to move that to a different location, say for dispensing, for example, and you can actually move the block and plate together. And then the final one, just showing a plate being moved to a shaker position, which is below deck. So this gives you a view of the below deck capabilities. So you see where the plate is going is to the shaker. Just in front of that is a incubated plate position. So you could move a plate there for heating or cooling. And you can also move plates around with the thermal block. So you can move it from active cooling on the incubated position to passive cooling by moving the plate with the block to the deck. So there's a lot of versatility with the options available in the machine, and you can really move even heavy things such as the mag blocks, such as the cooling blocks, with this versatile gripper. So that's now just moving the plate back to the deck. So I'll just move on briefly to the software. So we spent an awful lot of time developing the software to make it a very accessible and easy to use user interface. It's got secure logins, and you can log in at different user levels. So you can have standard users, you can just access and run protocols, and advanced users who are actually responsible for programming and editing those protocols. There's a degree of what I'd call intelligent programming, so you don't have to tell it every single move. For example, if you are doing a bead cleanup on a magnet, it will assume that that's done in a magnetic position, and it can move the plate directly to that position, and so on. So there's things that the software will do, and movements within the protocol, that just make it easier to actually program. It's also very graphical, easy to follow. You can see the screenshot at the top where it just shows exactly what's happening from one plate part of the process to the next, reagent dispensers and onto mag blocks and so on. So it's very visual, and we've found that users get to grips with it very quickly. It's also open license, so you can install it in any PC, and that means you can essentially alter protocols where you like. It could be in the coffee shop, at home, and then just send across the cloud or across a network the protocol to the machine for running. There's also powerful version management available, which allows users to look back to previously used versions and roll back and so on. The cloud is also quite a novel feature. So it allows, I've already mentioned, authoring protocols anywhere, but also sharing protocols across systems. So that could be within a site or across multiple sites. It really does help deepen collaboration within those sites, or within companies, or outside and between different companies or different organizations, or even globally. So there is a public element to this, where you can post protocols to the cloud for anybody to share, but ultimately you decide how much and how far you want to go with that. So the things that you can share, things like protocols, labware, liquid classes, and so on. So we're really hoping that this, as we increase the number of units in the field, is going to really broaden level of collaboration between our users. So just quickly wrapping up the key benefits. We've got great combined functionality for pipetting, dispensing, incubating, and shaking, and it overcomes the need for multiple instruments. So it's saving cost and, importantly, space, and it really does reduce the training burden with the ease of use of the system. At this moment, we've really focused on library prep and a few other things like PCR setup and so on, but it's a dedicated instrument which has been developed with the needs of those applications in mind, to develop to provide good accuracy and better performance for those applications. The compact footprint we're finding is very well received because people do have issues in the lab with space, and generally our users can find space for the Firefly versus other systems that may be much larger. We believe it sets a new standard for intuitive collaborative software, and it provides easy access to a range of applications and protocols and allows users to share those protocols. And the ease of use means that you don't need experts in the lab necessarily to run this. It should be something that's accessible for anybody. So at that point, I'll hand over to Maryia, who's going to talk through and introduce the application that we're talking about in detail for today's call.
Thank you, Paul, for introducing Firefly. I will now take over and walk you through the automation of NEBNext Ultra 2 FS DNA library prep on Firefly. Next generation sequencing library preparation is a complex process that involves multiple tedious and time-consuming manual steps, which makes it an ideal candidate for automation. The automation of NGS library preparation offers several advantages, including increased efficiency and improved consistency and reproducibility. However, automating the library preparation process requires a reliable and user-friendly liquid handling platform that can perform all the protocol steps accurately and precisely. Here, we present a solution to this challenge by demonstrating that our Firefly, a compact and innovative liquid handler, can successfully generate high-quality libraries for next-generation sequencing. This is exemplified by our automation of NEBNext Ultra 2 FS DNA library prep. Let me draw your attention to the capabilities of Firefly that make automated complex processes, such as NEBNext Ultra 2 FS DNA library prep, possible. The Firefly contains 2 moving decks with a total of 16 deck positions that can accommodate SBS footprint labware and accessories, such as magnetic blocks and thermal blocks. It contains 2 liquid handling heads, an air displacement pipetting head, and a non-contact positive displacement dispensing head. The air displacement pipetting head is comprised of 384 pipetting channels and can aspirate and dispense from both 96 and 384 well plates, depending on the format of the tip array that is presented to the head. The positive displacement head can dispense up to 6 different reagents to 96 and 384 well plates on the deck. We have a gripper to move labware and accessories during the protocol run as required. We also have a temperature-controlled position for reservoirs for the dispensing head reagents so that the reagents can be kept at optimal conditions during the run. There's a separate heater and shaker module for plates. We also refer to them as process modules. Now I will move on to describe the implementation of NEBNext Ultra 2 FS DNA library prep on Firefly. The NEBNext Ultra 2 FS DNA protocol was split into 4 sub-protocols for the purpose of automating them on Firefly. And these sub-protocols were sequentially run on Firefly. The first sub-protocol is called fragmentation and prep adapter ligation. The second one is the adapter cleanup. The third one is the PCR. And the last 4th one is PCR cleanup. On the slide, you can see the approximate on Firefly runtime for each of the sub-protocols. Between the sub-protocols, the user needed to perform some operations, such as replenishing reagents, syringes, and pipetting tips. Also, some of the sub-protocols included off-deck processes on the thermocycler, and there were also some thermocycler processes in between the sub-protocols. Let's take a closer look at a number of key points of the NEBNext Ultra 2 FS DNA prep that were employed on our Firefly protocol. In contrast to NEBNext Ultra 2 DNA prep that relies on physical sharing of genomic DNA or other DNA inputs, NEBNext Ultra 2 FS DNA prep uses an enzymatic transposase-based method to fragment DNA. In this protocol, DNA fragmentation and end prep are performed in one reaction, and the end prep includes end repair and the DA tailing. We use NEBNext adapters that have a hairpin loop structure and contain a uracil base. This uracil is removed by user USER enzyme in order to open the loop and enable subsequent PCR steps. User stands for uracil-specific excision reagent. This protocol was streamlined by implementing certain modifications that are used in the ATIC-NEB SARS-CoV-2 protocol. In particular, dispensing of user and PCR reagents was performed in the same step, and these reagents were premixed. The subsequent incubation steps were also combined. In terms of the setup for the NEBNext Ultra-2FS DNA prep on Firefly, we used 100 nanograms of human genomic DNA as input per sample. This genomic DNA was obtained from Promega. For certain steps, we use specific protocol variations, as the generic NEBNext Ultra-2FS protocol allows for variability in some steps. The ones we used are in particular the fragmentation and prep incubation time. This protocol was set as 30 minutes at 37 degrees, followed by 30 minutes at 65 degrees. For adapter cleanup, we did not use size selection, and there was just a single bit addition at this step. In terms of PCR cycles, we limited it to 4 cycles, in line with the recommendation from the kit provider. As for the labware and consumables, we used an Alpaca Magnum FLX magnet that you can see on the slide on top. It is a 96-well ring magnet. As for the plates, we used 96-well PCR plates. You can see a picture below the magnet on the right of the slide. These are Eppendorf twin-tech plates. For our run, we prepared 48 samples, which corresponds to 6 columns on the plate. And this included the no template or NTC controls that did not have any DNA input. The assessment of the quality of DNA libraries is, of course, an important quality control step that needs to be taken prior to loading materials into sequencer. This evaluation also underscores the efficiency of the library prep protocol. And we performed this QC step during our run, or rather after our run. Following the library prep on Firefly, samples were diluted 1:3 in 1x buffer, and then they were run on a fragment analyzer 5200 from Agilent, using a high sensitivity NGS fragment kit, also from Agilent. Fragment analyzer is a capillary electrophoresis-based system that is commonly used for quality control of nucleic acid samples, including DNA libraries. The system is used to assess the size distribution and quality of DNA libraries, as well as to quantify DNA in the sample. As consumable use is an important consideration for automating protocols, I will summarize the consumable requirements for running NEBNext Ultra-2FS DNA prep on Firefly. The number of consumables I'm quoting below is based on no reusing of pipetting head tips and no reusing of dispensing head syringes, with the exception of syringes that were used for dispensing of 80% ethanol. We also used genomic DNA and indexes supplied in a 96-well plate. And given these conditions, as you can see from the table on the slide, we would require 14 pipetting head tip sets, 12 dispensing head syringes, and 12 dispensing head reservoirs for reagents, as well as 3 empty 96-well PCR plates and 1 empty 96-well plate for waste. Next, let's zoom into the steps that make up the NEBNext Ultra-2FS DNA protocol on Firefly. In the next couple of slides, I will show you tables that detail the steps that were executed on Firefly in order to process samples through the NEBNext Ultra-2FS DNA workflow. As runtime is quite an important consideration for protocol automation, I will highlight the time taken to run groups of steps and sub-protocols for this protocol. The total time taken to execute the entire workflow from beginning to end was about 4 hours. Now let's take a look at the first sub-protocol that includes fragmentation, end prep, and adapter ligation. This sub-protocol consists of Firefly instrument on-deck dispense of FS enzyme and buffer, pipetting of gDNA input from the source plate, and mixing on the shaker, followed by off-deck incubation in the thermocycler for the fragmentation and end prep. After this incubation in the thermocycler, the working plate is returned into the Firefly instrument for on-deck dispensing of adapters and ligation mix plus enhancer, and for shaker mixing. This is followed by off-deck incubation in the thermocycler for adapter ligation. In this sub-protocol, the total time spent on Firefly instrument is about 13 minutes. The second sub-protocol is the adapter cleanup, which includes a single bit addition step and no size selection. This sub-protocol is run in the Fire instrument in its entirety. We were able to do that by leveraging the liquid handling capabilities of dispensing and pipetting heads, magnet compatibility, and the ability to incubate plates on the plate thermal module. As for the specific steps, the dispensing head is used for magnetic bead addition using the volume ratio of 0.8 volumes of beads to 1 volume of sample, followed by tip mixing and incubation on thermal module at 23 degrees for 5 minutes. After this incubation, the working plate is moved to the on-deck magnet, and supernatant removal and 2 wash steps with 80% ethanol are performed. The plate is moved off the magnet for dispensing of the elution buffer, followed by tip mixing and incubation on the thermal module. The eluate is then aspirated from the plate on the magnet to complete the adapter cleanup sub-protocol. In this sub-protocol, the total time spent on Firefly instrument is approximately 40 minutes. The third sub-protocol is called PCR, and it includes dispensing of the PCR master mix and user in a single dispensing head step, mixing on the shaker, and the addition of indexes using the pipetting head, which is followed by mixing on the shaker. All these steps are performed inside the Firefly instrument, and in this sub-protocol, the total time spent in the Firefly is about 10 minutes. After this, the working plate is moved to the thermocycler for the PCR program. The final sub-protocol is the PCR cleanup. This sub-protocol shares similarities with the second adapter cleanup sub-protocol, and it can be performed entirely inside the Firefly instrument in approximately 40 minutes. Individual steps include magnetic bead addition, this time using the volume ratio of 0.9 volume of beads to 1 volume of sample, followed by mixing and incubation on the thermal module. After the incubation, the working plate is moved to the on-deck magnet, and the supernatant removal and 2 wash steps with 80% ethanol are performed. The plate is then moved off the magnet for the elution buffer dispensing, followed by tip mixing and incubation on the thermal module. The eluate is aspirated from the working plate on the magnet and moved to the final destination plate, and that is the last step of our protocol. Now we completed the walk through the NEBNext Ultra 2 FS DNA workflow, and I hope you're convinced that this protocol can indeed be efficiently automated on Firefly. However, the crucial question remains: how good are the libraries that were prepared on Firefly? To answer this question, I would like to share some QC data on the libraries obtained from NEBNext Ultra 2 FS DNA prep on Firefly. We processed 48 samples, including non-template controls on Firefly, and the resulting libraries and controls were diluted in TE buffer and run on the fragment analyzer. I will show you the fragment analyzer output that is used to compare the distribution of fragment sizes for each of the libraries and the controls. On this slide, on the left, you can see the graph corresponding to the fragment analyzer traces for 42 libraries prepared on Firefly. On the right, there's a graph that corresponds to the no template controls for wells that were processed without any input DNA. Both graphs show the distribution of DNA fragment sizes in a sample, with the x-axis representing the size of the fragment in base pairs, and the y-axis representing their abundance. The left graph clearly shows distinct peaks, suggesting that the 42 libraries possess a consistent distribution of fragments that have an average size of about 350 base pairs, which is in line with anticipated library sizes. In contrast, on the right graph, there are no comparable peaks in the area of the graph corresponding to the expected library sizes, which indicates that there is a lack of detectable contamination in the non-template control samples. This means that Firefly performs consistently and does not show any cross-contamination between wells. To assess the uniformity of library prep on Firefly, we can compare the average library size and concentration across the test samples. The bar charts on the right of the slide represent the data from Fragment Analyzer for individual libraries, and they highlight the mean, range, and percentage CV for average sizes on the top graph, and for concentration on the bottom graph. As you can see, the 42 libraries prepared on Firefly using NEBNext Ultra 2 FS DNA prep protocol have a mean size of 342 base pairs, with a range of 41 base pairs and a percentage CV of 2%. These libraries have a mean concentration of 24 nanograms per microliter, with a range of 7 nanograms per microliter and a percentage CV of 6%. These results indicate that libraries prepared on Firefly show really good sample-to-sample uniformity, which means that the samples are treated equally across the whole plate. In summary, the results I've presented demonstrate the successful application of Firefly in executing an NGS library preparation workflow as exemplified by NEBNext Ultra 2 FS DNA prep. The libraries produced on Firefly are uniform in concentration, and the distribution of fragment sizes is uniform across the samples. There was no detectable contamination found in the control wells that were processed without any DNA input. The successful automation of NEBNext Ultra 2 FS DNA prep protocol is just the start of our efforts. We're collaborating with various reagent and technology providers to create automated NGS workflows that cover a diverse range of applications. The names listed on the slide on the left are just a selection of the protocols we're currently developing. So keep an eye out for upcoming automated protocols on Firefly. Thank you very much for joining us today, and we're happy to answer any questions you have on Firefly and automating your NGS preps.
Just before we move to questions, I'd just like to take the opportunity to alert you to some opportunities to see Firefly in the flesh. So we are taking Firefly on tour in the US. You can see the dates on the right. So starting soon from the 15th of May, we'll be in Cambridge, Massachusetts, and then moving through to New Jersey, onto Philadelphia, Baltimore, and Raleigh, North Carolina, and finally to Texas in June. We're also in the process of finalising dates in Europe. So watch out for those dates and locations. That should be happening around June time across a variety of cities in Central Europe. And we're also going to be at the SLAS EU conference at the end of May in Brussels, where again we will have the Firefly. So lots of opportunities to see it for real if you want to get a better feel for it, and please join us on one of those occasions if you can. You can scan the QR code, and that will take you directly to the link on our website for the Roadshow tour. And with that, we can move forward to questions.
Thank you very much, Paul and Maryia, for a very interesting presentation. It is now time for a question and answer session, and just a reminder that if you do have any questions for our speakers, there is still time to send those in. You can do so by using the box to the right of your screen. Right, so let's get started. The first question here I've got asks, With non-contact dispensing of viscous liquids, can you guarantee the accuracy of small volume dispensing? And will the user have the ability to modify the speed of aspirating or dispensing viscous solutions?
Yes, our core technology for non-contact liquid dispensing is largely liquid type agnostic, which means that it can handle a broad range of different liquids, including very viscous liquids and even volatile liquids. We generally don't have to do too much adjustment to it. However, all these options are exposed to the user, so you can adjust the speed, you can adjust quite a few different parameters. And we also have what's called liquid classes, which have optimal settings for specific reagents, such as viscous reagents. And just as a side note, our non-contact dispensing technology was used quite a lot during COVID to dispense complex liquids such as master mixes. So it's well proven.
Great, thank you very much. The next question I have here asks, is 48 the maximum libraries you can prepare per batch?
No, you can prepare a full plate, or you can prepare as many samples as you need. In our particular setup, that's how many samples we had, but you absolutely can prepare a whole plate of 96 samples, or you can prepare a whole plate of 384 well samples, or you can prepare specific columns on your plate as desired.
Great, thank you. Next question here is asking, after initiating a protocol, is there an option to stop and restart the Firefly at different stages?
Yeah, so it's very easy to actually, within the execute part of the software, you can actually jump into the protocol at any point. So if you had need to stop the protocol, there is a stop button. You can stop it, and then you can restart it at your chosen point if you wanted to go in and manually manipulate something, for example. You can also pause a protocol if you want to interact. If there's some sort of issue that you need to deal with as well, you can stop the protocol, you can deal with that issue, and then you can start it again at any point. So it's actually really easy to jump into the protocol at any point that you want to.
Great, thank you, Paul. Next question asks, with the run setup, have you seen similar results with less DNA? So for example, if you use 25 ng human QG DNA instead of 100 ng?
We haven't run a prep with 25 nanograms. However, we're quite confident that that should be possible. The kit itself, the NEBNext Ultra 2 FS, allows protocols for input of under 100 nanograms, over 100 nanograms, and for larger fragment sizes. And the protocol we've been running is very close to the lower input protocol. So I think it should be possible. However, we do not have data on that yet, as we've focused on optimizing this protocol for 100 nanograms in the first instance.
It's probably fair to add as well that with our other platform, the Mosquito, we've spent an awful lot of time miniaturizing applications where typically you're working in like a tenth of the volume. There's certainly some scope to miniaturize applications on the Firefly as well. Perhaps not as far as we do on our other platform, the Mosquito, but that might be another approach.
Fantastic, thank you. And the next question asks, what is the optimum input DNA quantity for library prep?
For this, it really depends on your application and on your DNA. At the moment, there are a lot of different kits available on the market that address very low input volumes, as well as different DNA types from different organisms, for instance. And in terms of automating for Firefly, if you can do it manually, we can automate it on Firefly.
Great, thank you. And next I've got an attendee asking, does the Firefly come with barcode reading on plates to differentiate the plates loaded onto it? Or is there a way to record which plates have been loaded and run on the machine via the software or a cloud?
Yeah, so firstly, the actual reading of barcodes. There is an option to supply a barcode scanner that you actually use as you load the plates onto the system. So essentially it's a hand scanner. So as you load the system, you scan the barcodes, and within the software you can select to record those barcodes and then provide them in the output reports. So if you wanted to kind of capture that information to LIMS, for example, you can do that. So yeah, essentially you can track the actions and which barcoded plates were on the system and send that data out.
Great, thank you. Next, this question asks, is the machine designed to run the protocol automatically, or do you need to write the code or program?
Quick answer to that is both, actually. So for example, the one we presented today is available to download, so users can download that to their own machine and run it. Our internal applications team that Maryia is a part of are developing new methods all the time, so they're working on a number of projects at the moment, and as they test those and go through QC, they will also be added to the cloud for users to download free of charge. But the software is also very easy to program. So if a user wants to develop their own applications, they can also do that very easily and very quickly. And I think one of the really nice things is because particularly with the dispensing technology, there's no real fine tuning that you have to do there, unlike what you have to do with the pipetting side of the system. So as a lot of kits are additive, just adding various reagents like enzymatic reagents and beads and so on, you can actually build those protocols up really quickly without any kind of expertise or fine tuning of the liquid handling, which makes it very quick.
Fantastic, thank you. Next question asks, is it possible to adjust the protocol to use half of the reagent volumes?
Yeah, I mean, what I've just kind of said kind of alludes to the answer there. Yes, you're completely free to develop the protocols. There are also variables that can be used. So if you wanted a kind of standard protocol, you could set the volumes as variables, and you could link the different. So for example, a starting transfer volume could have a variable that then is linked with other variables in the protocol. So if you then decide to use half the starting volume, it will half everything else. So you can actually make your protocols quite flexible with just a simple user input at the start to say that you're now going to do it a half volume or a quarter volume. Obviously, the only thing to be mindful of there is kind of working within the dynamic range of the system.
Great, thank you. Next question here asks, does the system have liquid level sensing?
No, it does not, because you're using a 96 or 384 array or columns of 8 or 16. Liquid level sensing requires kind of individual control of each of the tips, which isn't possible in a multi-channel head like that. However, what we have done is the labware that we've mapped. We've used a system called a volume check that we have that allows us to actually define the profile of wells. So things like V well and U well plates can be quite difficult to get the exact profile. So we've done standard curves in those from first principles, where we've pipetted from low volume through to the maximum volume, and then we've mapped that with our volume check system, and that profile is within the software. So you can use liquid tracking, and you can use things like liquid tip touch, which are based on the volume that the software knows in the well from your protocol against the mapped well profile that is in the labware file.
Fantastic, thank you. I've got another question here that asks, do I need to be at the machine to write protocols?
No, you don't. The software is open license, so you can load it onto as many Windows PCs as you like. If you use Macs, it will work if you use Parallels, which is the Windows emulator software. But essentially, you can author protocols where you like, at your desk, in the coffee shop, wherever, and then you can send them directly to the machine over the cloud. You could send them across your network. You could even put it on a USB stick and get it across the machine that way. So there's various ways that you can get it to the machine.
Great, thank you. Next question asks, can the incubator be used for PCR?
No is the simple answer to that. It's there's a couple of things. So the incubated plate positions really are for incubation. So it's programmable within protocol. You can change the temperature of it within a protocol. So you go from hot to cold, but it doesn't. One, it doesn't cycle through temperatures fast enough, and two, it doesn't have a heated lid. With the current Firefly, PCR reactions will be off deck. However, we are developing and will be launching next year the Firefly Plus, which is an expansion unit for the Firefly. So it will attach to the side of the Firefly, and it will contain an on-deck thermocycler, and will have the ability to take plates from the Firefly into the thermocycler, add a sealing lid, so an automated sealing lid, run the PCR reaction, and then remove the lid and put the plate back to the Firefly. It'll also have extra tip capacity because as you then go end-to-end workflows, you need more tips to actually to run that full workflow. And that will be it'll be possible to add that to an existing Firefly, so customers who want to add that later can do.
Fantastic, thank you. Got another question here asking, is it possible to use variables in the protocols?
Yes, we do have an option to use variables. These variables are used at quite a high level in the protocol, so they will apply to all the steps that are relevant. One common case of using variables is for the number of columns you're running, which would correspond to how many samples you have. So you can define as, say, 3 columns, and then next time you want to run your protocol, you can change it to 12 and run the whole plate. Alternatively, you can use variable to define, for example, the file path for a CSV file that you can use for defining your dispense in the plate for applications such as normalization.
Fantastic, thank you. I've got another question here that's asking, when preparing 96 or 384 libraries at a time, how many additional pauses for tip refills are needed, and what would be the runtime impact?
So that very much depends on the protocol, and it varies from kit to kit. I mentioned at the moment that the PCR steps would be offline. So typically we find that the protocol is walk away between PCR, and then you can replenish the deck. The time is very quick. I mean, you can change all the tips out in probably less than a minute. It's very quick to do that. But don't forget, you can also you're really minimizing the amount of pipette tips you use, because all the reagent addition steps we tend to do on the dispensing system with the multi-use syringe. So you would dedicate a syringe to each reagent type that you're using, and that massively reduces the number of tips that you use. Going forward, as I've already mentioned, as we move to the Plus system and the on-deck thermocycler, then that will have extra tip capacity and we'll be able to replenish tips automatically.
Fantastic, thank you very much. I think that's all we have time for today, but I would like to say thank you once again to Paul Lomax and Maryia Karpiyevich from SPT Labtech for joining us and for today's really informative webinar. And thank you to everyone who joined us online today and for sending in all those fantastic questions. I hope you all found this to be a really worthwhile session. If we didn't have time to answer your question, we will follow up with these after today's event. Or if you have any other questions, you can email me at editor@selectscience.net, and I can follow up with your questions for our speakers. You can also download a certificate of attendance for today's session. You can find that in the Related Resources tab on your screen. And if you'd like to listen again to today's webinar, or maybe invite a friend to listen, it will be available to watch on demand in just a few days' time. So for now, goodbye, and thank you once again for joining us.